Okay, as promised, I'm making another video for the Unit 5 review. You're in my guest bedroom yet again. Welcome back. It's a bedroom for guests. That's what you are. So, hi. I'm going to try and make this video like one big video. I'm going to see if YouTube will allow me to do that. If not, I'm going to cut it into 15-minute sections if YouTube is being mean. Okay? So, what we're going to cover first is cell division. Okay? There's two types of cell division: mitosis, meiosis. Okay? I'm going to start with mitosis because that's the one we started with, and I would like to recover that because it's the material that's the oldest and it's the less of the two. It's less fresh in your minds. So we're going to cover mitosis first. So I'm going to walk through this real quick. So mitosis. Mitosis is one part of the cell cycle. Okay, so the cell cycle has three parts to it. The first part is interphase. The second part is mitosis. And the third phase is cytokinesis. Okay? So we're going to tackle each of these one at a time. Okay? So what I want to tackle first is interphase. What is significant about interphase? Well, the cell does a couple of things during interphase. Okay? During interphase, the cell grows. It's one of the main things it does. Okay? I'll explain later why it grows specifically. So it grows. In order to do mitosis, in order to do any cell division, okay, cytokinesis, you need to replicate your DNA. Okay? You need to replicate your DNA because we need all of our cells to have exact copies of all the DNA. So you need to make sure you have two copies. So when you take these two copies that you made, and now you're going to separate into two daughter cells, each daughter cell is going to end up with a copy. So we need to do DNA replication. Okay? And last but not least, in interphase, your cell is doing its job. Okay? Whatever cell it may be, if it's your stomach cell, it's going to be taking in nutrients that you're putting into your stomach. If it is your cells in your lungs, it's going to be exchanging oxygen from the air and also releasing CO2 so we can exhale that. All of your cells have a specific job. They have to do the job. And interphase is the longest phase. Okay, the cell spends about 90% of its time in interphase doing all these things. And that makes sense because you want your cell to be doing its job most of the time, because if most of your cells are not working and doing their jobs and working toward a common goal, which is keeping you alive, you're not going to be alive for much longer. So interphase, 90% of the time, grows, cell does DNA replication, and the cell does its job. I'm going to erase that. Next, we're going to talk about mitosis. And when I'm talking about mitosis, I'm going to be drawing the phases out. I'm going to try to also, I don't have multiple colors, only have black, so I'm going to try and do my best to show multiple different chromosomes. Okay, so mitosis consists of four phases: the P phase, the M phase, the A phase, and the T phase. Okay, P mat, prophase, metaphase, anaphase, telophase. Okay, so how do these phases look different? Well, if I were to do interphase at the very top, what I'm going to see is a cell with a nucleus on the inside, and the DNA is kind of just all up in here, unraveled, just filling in the entire nucleus everywhere. Okay, cell with a nucleus. Okay, and the DNA is all unraveled. We need to start condensing that DNA so we can start moving it around. Because if you remember correctly in class, when I took that big string of yarn, I started moving it around with another student, we were hitting everybody. And then somebody ended up snipping it with some scissors. That's not good for the DNA. We don't want to damage our DNA when we're moving it. So we need to compact it down, roll it down, coil it up, so we can move it around more easily. So in the P phase, so this is interphase, this is the P phase, prophase, we're going to have our nucleus, and it's going to start disappearing. So I'm going to represent that as a dotted circle going around. It might be a little hard with the light glare, but that's okay. So dotted circle, and our chromosomes are going to start forming X's. So I'm going to do one X here. I'm going to kind of do a squiggly X to make this X look a little different. So one normal X and then one squiggly X. Okay, for you guys, it kind of looks like a bold X. That's fine. A normal X and a bold X. Cool. Okay. What happens after that? Well, the nucleus is going to disappear in metaphase, the M phase, and the chromosomes are going to line up in the middle. So we're going to get our one chromosome up top. And we're going to get our fuzzy, bold, squiggly X lined up in the middle, just as so. Anaphase is the away phase. So what we're going to do is we're going to have our cytoskeleton on each side of the cell. They're going to reach out and they're going to start pulling those X's apart. So we're going to have individual lines of chromosome being pulled in opposite directions. So each cell gets one copy of each chromosome. And then telophase, we're finally going to have the two cells almost separated. We're going to have two nuclei starting to form. And then our chromosomes are going to be in line structure. Line structure, not X's. And the cells are almost divided. Okay? The easy way to keep track of all of this, okay, is you start with X's. Anytime you start with cell division, you're always going to start with X's. Whether it's meiosis or mitosis, you're going to start with X's, okay? Then you're going to make your way, and then once you pull these X's apart in mitosis, they're going to be individual strains, okay? Individual. So right here, I have one line, one chromosome. I have a fuzzy one here. I have one chromosome and then another fuzzy one, okay? So each cell got one copy of each chromosome, okay? So that is PMAT, that's mitosis. Sorry, the glare is, again, not amazing. Apologize. So that's mitosis. What is cytokinesis? Cytokinesis, last but not least, is when the cell from telophase, so we had our telophase cell that looks something like this. Our two different chromosomes. Cytokinesis is when those cells actually form into two small daughter cells, and now they're going to go straight into interphase, and all their DNA is unraveled all over the nucleus, okay? This is called the cell cycle because when you do interphase, you're going to go into mitosis. From mitosis, you're going to do cytokinesis. Once you're done with cytokinesis, you're going to go all the way back to interphase, and you're going to keep doing this in loops and loops and loops if your cell is healthy, okay? Now the question is, why do we grow in interphase? It's because when you produce your two new daughter cells, well, if you take one really big cell in the beginning, and then you split it into two cells, these two cells are going to be smaller, okay? They're going to be essentially like children's cells. They need to grow up so they are like parent cells, full grown. So that's why in interphase we do the growing. So that covers a lot of mitosis and some of the basics of the cell cycle. What I would like to cover next, before I hop into meiosis, is your chromosome structure. Because this is something that we practice with a little bit, but I think people would kind of get this messed up if I give you a test on it, let's say on Thursday. Crazy. If I use this video for future classes, your test may not be on Thursday. Let that be known. Okay. This is recorded on March 4th, 2025. Okay. So, chromosome structure. I'll just do struct. Chromosome structure. If anything, I can also talk about DNA right now. That's crazy. Okay. So, what is a chromosome? Well, a chromosome is a condensed version of your DNA. So if I took your DNA and I did this and this, and I made all the bonds here. So this is DNA. If I start taking your DNA and I start coiling it up and tightening it up, soon enough what it's going to make is a big old line like this. If I keep coiling it up, this is essentially a chromosome, okay? We take the DNA and we tighten it all up, and as we bundle it up over here and we keep bundling it up, we make a chromosome, okay? For simplicity's sake, instead of having a big scribble right there, I'm just going to redraw this as a single line. Easy enough, okay? So when you're talking about a single bundled up DNA strand, that is called a chromosome. Sorry, my whiteboard is bouncing off the wall. Chromosome. When you're talking about one bundled up strand of DNA, bundled up molecule of DNA, there's a couple other terms that go with this, okay? As I said earlier, in interphase, you have to replicate your DNA before you go into mitosis, okay? You have to do DNA replication. So how does that look? If I want to replicate this DNA, well, let's say I do. So let's say I do bigger arrow, DNA replication. What I'm going to get is 2 lines. Easy enough, okay? Get 1 chromosome, replicate it, you get 2 chromosomes. Seems pretty easy. But what we're going to do with these 2 chromosomes is we're going to attach them and form a structure out of them, okay? So we're not going to have them as individual chromosomes. When we start moving them around, we want them in their X shape. Now, many of you draw your X's like this. That's an X. Yeah, you're not wrong. And they get attached in the middle by a little piece here that we'll go over. But that's not 100% accurate. That's not actually how it looks. I know your popsicle sticks that we use in class look like that, but this is actually how they look. So that's wrong. I'm going to do the more accurate chromosome structure. What really happens is one of your chromosomes, let's say the original, this one here, let's say this one actually bends and kind of has like a C shape to it, almost like a C shape, like a reverse C. The new chromosome that you make is going to also have a C shape to it, and they're going to kind of bend toward each other, and then in the middle you're going to attach them. So realistically, if you, I'm gonna like star pose. If I'm an X, I'm an X. This arm and this leg are actually part of the same chromosome. All through here is the same chromosome. All through here is the same chromosome. So really, the same side, the same side is the same chromosome, okay? So the same arm and leg are the same chromosome. These are exact copies of each other. So what we call these 2 chromosomes together, since they're exact copies, we call these sister chromatids. Sister chromatids. Okay, we're using that terminology of the female familial terms. Sorry, because for example, in mitosis, we produce 2 new daughter cells. There's no such thing as son cells, so we produce 2 identical daughter cells. When you make an exact copy, an identical copy of a chromosome, we're going to use the same term, but instead of daughter, we're going to use sister chromatids. What is this piece right here that's connecting them? This is called the centromere. What that does is it connects your 2 sister chromatids so they stay together. Okay? Chromosome by itself. When there's 2 chromosomes and they're the exact same, they're just copies of each other, sister chromatids, and they're attached by a centromere. There's one last type of chromosome I need to go over. You have 2 sets of all of your chromosomes. And the reason you have 2 sets is because all of you guys came from 2 parents. Okay, 1 mom, 1 dad. So let's say I'm talking about this chromosome. Okay, normal straight chromosome. Let's say I'm talking about this other one that kind of looks a little fuzzy. I'm going to say this is my dad's chromosome because my dad's a fuzzy guy. Hairy man, I'm saying. He's a hairy feller. Okay, this is my mom's chromosome. Mom. This is my dad's. When you're talking about your mom and dad's chromosomes together, these are the same chromosome, but this one came from mom, this one came from dad. They're the same chromosome, but they just came from 2 different people, your 2 different parents. We call these homologous chromosomes. So they're homologous. Even if you do DNA replication, if you do DNA replication and you get your doubled amount, so you get your normal mom X and then your dad's fuzzy X, fuzzy, very fuzzy, very fuzzy. And then I'm going to put the big dot in the middle. These are your centromeres. Okay. When you're talking about these two together, they're still homologous. You just have two, or I shouldn't say two. I should say you have a copy, two copies of each of your parents' chromosomes. When you talk about your mom's chromosomes and your dad's chromosomes together, they're still homologous. That's fine. As long as you're talking about both copies of your mom and dad's chromosomes, homologous. These two chromosomes on your mom are sister chromatids because they're the exact same. And these two on your dad are also sister chromatids because they're also the exact same. They're exact copies of one another. But if you're talking about the fuzzy chromosomes compared to the normal smooth chromosomes, then it's homologous because that's my mom and dad's chromosomes together. Okay? Chromosome terminology. While we're at it, we're talking about chromosomes and they're made out of DNA. How about we talk about DNA terminology? Again, more accurate DNA. Okay? More accurate. So I'm not going to do that twisty thing with the twisted ladders. Bad. We're going to do something else. More accurate. So there are three parts to your DNA. Your DNA is made of three main parts. So the first one is a sugar. And this sugar is called deoxyribose. Number two, nitrogen base. That's your A, T, G, and C. And number three, last but not least, you have your phosphate. Okay, these are the three things that make up your DNA. You guys in the lab building a DNA molecule, and we made out of candy. You guys made a lab report on it. Some of you did awesome, and some of you didn't do awesome. So we're covering it. Okay, I'm gonna move my backpack out of the way. Hey, that's better. So, how are these put together? How does the structure look? I'm going to try to do both strands of the DNA. So if you look on DNA, there's one strand that goes on up and down on one side, another strand that goes up and down on the other side. So I'm going to show both. So this might be the moment where you might skip ahead in the video, go 2x speed, or just skip until I start yapping and looking at the camera again. I'm going to start making this. I'm going to start describing it right in this space. So this might be the time to move ahead in the video, just to save you time, just to be considerate. See, if you skipped this part, you would be missing out on all my sound effects. Yeah, that looks pretty good. Yes, nice. I should scream randomly if you're scrolling through, so then, like, your parents will hear you, like, watching this video at night, and then they hear you scream, and they think you're watching a horror movie, and then they'll come in and be like, I thought I told you to go to bed, and you're like, I'm studying, and then they're like, no, you're not, and then you get grounded. I'm not your enemy. I'm a villain, not a monster. Okay. I'm just yapping. Okay. Super accurate DNA molecule right there. Some of the glare is not helping. I'm gonna stand on this side and see if it helps. Just a smidge. Just a smidge. So what are these circles? Well, I'll make a key. These circles have a P in them. That means these are your phosphates. These are your phosphates. The pentagons I drew, these have an S in them. These are your sugars. And last but not least is your boxes. I didn't put anything inside these yet, but these are your bases, your nitrogen bases. Okay? You have 4 nitrogen bases, A, T, G, and C. I am also making this accurate. I got some dots in the middle of here. You might not be able to see them. Maybe I'll make them a little bigger. Okay, they're a little bigger. So I have two bonds in between here. Not that you need to know this, but your A's and T's bond together. That you do need to know. But the reason they bond together is because they have two weak bonds that hold them together. Okay, the two bonds aren't super important. I'm just telling you that just for more information. But down here, we have three bonds, so it can't be an A and a T because A and T's have two bonds. So this must be your G and C. Your G and C bind together. Okay. With this being said, your phosphates and sugars are always on the outside strand of the DNA. They're on the outside, and I made super solid lines between each of these sugars and phosphates because these are strong bonds. Okay, strong bonds means they can hold up and they can get protected. Not get protected. They are doing the protecting. What they're trying to protect, these sugars and phosphates on each side with their strong bonds, what they're protecting is the code in the middle. Okay, your A's, T's, G's, and C's, that's your genetic code. It codes for everything that you are, and you don't want that getting damaged. You don't want your bases in the middle getting damaged. Because if they get damaged, that's called a mutation, and many mutations don't actually do anything, but there's a small chance they could do something good, but there's more likely of a chance they're going to do something bad. Okay, so we don't want our code in the middle getting damaged. That's very bad for us. As well as, I would like to highlight these weak bonds yet again. Okay, we have weak bonds in the middle of our DNA molecule, right here, inverted controls. We have weak bonds. The reason we have weak bonds is we want the strands, these two strands, so we have one strand here, one strand here. We want them to stay together. We don't want our DNA to float off and then have our code exposed to the open environment. We want the code to stay in the middle, protected. So we want these bonds here, these weak bonds, hydrogen bonds. These are weak hydrogen bonds. But we want to be able to replicate our DNA whenever we want to. Okay? So when we try to replicate our DNA, it'll take an enzyme that will actually come through and break these bonds. And it can do it pretty easily because these are weak bonds. They're weak bonds. Weak things are meant, in a sense, to be broken. So we will cut down the middle of the DNA strand, between the middle of the two DNA strands, I should say, down the middle of the DNA molecule, and we're going to open up this code so we can actually read it and copy it. Okay? And then once we're done copying it, then we'll put these very weak bonds back in. As I show these weak bonds as dotted lines, not solid lines. Okay? And we'll put those weak bonds back in, put the DNA back together. Okay? So, DNA replication, the parts of the DNA. The runs of the ladder, so this kind of looks like a ladder. If I were to continue it, it would look like you can climb it like a ladder. Your runs of the ladder, your base, sugar, and phosphate all together are called one special thing. So when you have a phosphate, sugar, and base in the middle bound together, this is called a nucleotide. Nucleotide. And all a nucleotide is one half of a run. So here's a nucleotide here. Here's another nucleotide here. When you put these two nucleotides together, you get one run of the ladder. You get one step. Here's another nucleotide. Here's another nucleotide. Another step in your ladder, another run. Okay. So far, I've drawn 4 nucleotides on this ladder. I have a free phosphate up here, and I have a free one over there. I can make another nucleotide up here and right here too. Okay. And this goes on for millions and millions and millions of steps to your ladder. Okay. Your DNA is very, very long. DNA structure. We talked a little bit why the weak bonds are weak and how that helps with replication. The parts to the strand of DNA, why we want the code in the middle, why we want it protected, and what is it called when you change your DNA when you don't protect it properly. That is called a mutation. Lovely. Next, on the chopping block, do I want to cover cancer or meiosis next? Those are the last two. Cancer is a lot easier to talk about. Meiosis, not so much. So maybe I'll save meiosis to the end. Cancer. Cancer. what is cancer? It is, number one, first bullet point, uncontrolled cell division. Uncontrolled cell division. That's what cancer is. So that means cancer isn't spending much time in interphase. A cancer cell isn't really doing its job. It's not doing what's supposed to. It's not really taking a time to do what it needs to do to keep you alive. What it's worried about is doing nonstop cell division. So that means it's going to go straight into mitosis and do cytokinesis after it gets big enough, and it replicates its DNA. Once it replicates its DNA and it gets big enough, it's like, we're splitting it again as soon as possible. We're not waiting. And that's a problem, okay? Why is that a problem? Well, two things. Number one, my cells replicate and they divide, but they do that for a reason, okay? The reason any of your cells go through mitosis is really for two things. Your body does mitosis, cell division. Your body does cell division to grow, okay? I don't grow anymore. I'm a 23-year-old man, almost 24. I'm done physically growing, more or less, when it comes to like height, let's say. The other reason is if you get damaged. Okay, I get damaged all the time. If you were looking at my arm, I have a bunch of like white scratches and scars all over the place. That's because I play with my cat, and my cat has claws, and I have really tight skin, and then his claws just rip through my skin all the time. So I do mitosis in my arms all the time because I'm healing from my cat wounds. That's just how it is. You heal from injury and you're growing. Okay, that's why we do mitosis. And we don't do consistent growing because obviously if I was consistently growing and growing and growing, I would be humongous. I would be like, I don't know, like Godzilla. But as you can tell, I'm a 5'8", 145-pound man. I'm not very big. So why am I not growing more? Well, that's because my cells are told to stop growing. As well as once I kind of hit the limits, like for example, my muscles or my bones or really anything in my body, once it kind of hits a maximum space capacity, it's like, oh, my cells in my chest are growing and growing and growing. And it's like, oh, bro, we're hitting ribs. My cells are hitting ribs. Guys, we're running out of space. Stop replicating. We filled the room. So once you run out of room, your cell also tends to stop replicating. Okay, so space is an issue too. Cancer doesn't care about that. Cancer doesn't listen to anybody. Cancer thinks it's right. Okay, it's a narcissist. Sounds familiar to like a certain teacher, you know? It keeps on replicating nonstop. Doesn't care about space. Doesn't care about doing its job. It just cares about its one job now, which is doing nonstop cell division. The second reason that's a problem, because it's going to be growing and growing and growing, and that's going to create a mass inside you, is it's going to take up a lot of the nutrients from your body. So your organs need nutrients, okay? You have blood, rich blood that carries oxygen and glucose through it, and it's carrying that to your cells so your cells can do cellular respiration so they can get energy. But if a tumor takes up all your arteries and absorbs all that nutrients, all your organs are going to be starved. You're going to be deprived versus this tumor is going to be getting so much nutrients that it's going to be encouraged to grow even more, making the problem worse. So tumors are dangerous and cancer is dangerous because they steal nutrients from you. It's almost like having a second living creature inside you that's you, but it's not on your team anymore. Okay, so cancer is really dangerous in that manner. With that being said, we talked a lot in class about the causes of cancer. Causes of cancer. And you guys listed so many things. You guys listed like sunlight. So sunlight, aka UV. I just said UV light. You guys said smoking or vaping. You guys also said it depends on your environment, where you live. Where you live can potentially give you cancer. If you're in a sunnier environment, if you're in an area with a lot of smog, heck, I mean, extreme example, but if you live in Chernobyl, if you live in the outskirts of that, a lot of radiation over there, might get cancer. You guys also talked about you can inherit genes from your family that are somehow mutated, and they're increasing your likelihood of getting cancer. Okay, nasty stuff. all bad things. An example of this is like, for example, if you ever go to a doctor and your doctor is concerned about your health in any specific way. Okay, you'll bring up, for example, like, oh yeah, my mom just got diagnosed with cancer. I want to check out if I have any chance of getting the cancer she got. Your doctor's going to ask you a few things. They're probably going to ask you, like, has anyone else in your family got that cancer? And you're like, oh yeah, my grandma and my great grandma, my aunt. And then your doctor's like, hold on, hey, now we got a humongous family history here. You might got cancer coming your way. Okay, that's really bad. Let's keep an eye on this. Let's keep an eye on you so we can try to catch it early so we can get rid of it. They also might ask you what you do. Do you smoke? The answer for all of you should be no, obviously, and to vaping too. You don't. That helps me sleep better at night. Don't smoke, don't vape. Or if you're out in the sun, you're not putting on sunscreen. And if not putting on sunscreen, well, might get skin cancer. Okay, if you tell your doctor that, they're like, well, you're kind of asking for it. So they might ask you things like that. These are things you can control outside environmental causes of cancer. You can't do too much about the genes, though. If you're born with a higher likelihood of developing a certain type of cancer because of genes, that's tough. Okay? Maybe sometime in the future we can change that, but as of now, if you inherit genes that can cause cancer, there's not too much we can do about it. What we can do about it is be aware of it so we can keep an eye on it for the future. Cancer. Now, last but not least, arguably the longest and the newest, freshest, and potentially hardest topic. Interesting, that one on the race. Meiosis. If you made it this far, congratulations. If I split this video up, then you don't know how much time it's been, but it's been almost 32 minutes. So very good. Very good job. So, meiosis, what's the purpose? We do meiosis to produce 4 unique gametes. Okay, that's a term, vocab word, gametes. Gametes mean sperm or egg cell. So when you're done with meiosis, you're going to take one cell, and you're going to turn that one cell either into 4 sperm cells, if you're a male, or 4 female cells, 4 egg cells if you're a female. My bad. So 4 sperm cells if you're a male, 4 egg cells if you're a female. Okay? One thing about this that's very special, you produce 4 unique gametes with half of the normal chromosome number. aka you get 1 copy of each chromosome. So you're going to form 4 unique sperm or egg cells, gametes, and they're only going to have 1 copy of each chromosome, half of what you usually have. Okay? And how's that half? Well, you guys have 2 copies of every single chromosome in your body because you have 1 from your mom, 1 from your dad. That's how it is. But we want to get down to only having 1 copy in each of the cells. So instead of having 2 copies like we normally have, you're trying to get down to 1 copy in each cell. Okay? So let's walk through meiosis. I'm going to erase this just for space so I can draw a little bigger. Okay? This is still meiosis. I'm just going to erase all this so I get a little bit more space. I'm going to try to make the chromosomes look a little different. Again, X chromosomes, maybe one's going to look a little bit more bold, one's going to look a little lighter. I'll do my best. So, meiosis is very much like mitosis, and that's a tricky thing because you might get those confused. Okay? So, watch this video a few times. Watch this part a few times. If you can get meiosis down, mitosis is going to be easy to understand. This is the hard stuff. Okay? So, the first part of meiosis is a process called meiosis 1, which is the first round of cell division for meiosis. So, we're going to do almost normal mitosis, almost. Okay? So, we're going to start with our PMAT. The first phase, our nucleus is going to be disappearing. I'm going to show, let's go with 4 X's. I'm going to do very bold X's. and I'll do normal X's. Bold X's, normal X's. Okay, that's a tiny X. These are tiny X's. Boop, boop. Actually, I'll make this one a little smaller. There we go, a little tinier. Next, we're going to line up our chromosomes in the middle, but this is the first difference in meiosis. Okay, usually you guys would line up all the X's, so they're essentially looking like this: 1X, 2X, 3X, 4X's. You line them all up in a single file line. That's not how it works in meiosis. In meiosis, what you're going to do is put the same exact chromosomes, okay? So the same exact chromosomes next to each other of the same size, okay? So for example, I have my bold chromosome here, which if you remember earlier, I said the bold fuzzy chromosome is my dad's chromosome. And I'm going to put my mom's chromosome next to it. And then again, I'll put my mom's smaller chromosome down here. I'll put my dad's fuzzy one actually on the other side, just to make it a little different. Okay? But here's a special thing that happens, okay? We want these 4 daughter cells, these 4 gametes, these 4 sperm/egg, we want them to be unique, okay? Right now, we're kind of following the same process a little bit. So this is how your cells make sure they're unique. You see these fuzzy X's as fuzzy chromosomes, and you see the normal chromosomes? What these chromosomes do, essentially, they go up to the chromosome of the same size, okay? So big chromosome, small, small. They'll go up next to each other and they'll swap a little bit of their chromosome, okay? Swap a little bit. So actually, my dad's chromosome up here, the big one, is actually going to get a normal piece that sticks out right there. My mom's chromosome right here is going to swap over her piece, and now she's going to get a bold piece right there, okay? Same thing with the small one. The small chromosome, one bold piece right here, and then down here, one normal piece, okay? They swap. That's called crossing over. I don't need you to know specifically when crossing over occurs in these phases, but I need you to know that it happens in meiosis 1. Meiosis 1, crossing over happens during prophase and the metaphase, okay? So prophase crossing over occurs, and now you can see it. You can see how a little bit of my dad's chromosome is on my mom's. You can see how a little bit of my mom's chromosome is on my dad's chromosome, okay? And you do that for both. We're going to separate the chromosomes, and when we separate them, they're going to be pulled apart, but they're going to be in their X forms. They're X forms, okay? So they won't be pulled away as individual sticks. They'll be still as X's. So here's one. And then here's this one. And here's the other one. Last but not least. So they're going to be pulled apart as X's, not as individual lines. They're going to stay in their X form as they get pulled away. And then what you're going to form at the end of meiosis 1 is almost 2 cells, and each cell is going to have 2 X's in them. And if you notice, none of these X's look quite the same to the other. And that's on purpose, because we want these cells to be unique compared to one another. We want them to be unique, okay? That's the goal. So this is the direction we've been going so far. Now here's the fun part, because now I get to do 2 sets of cell division for this part down here. So that's meiosis 1. That means we're going to move down here. And down here is going to be meiosis 2. If you notice, I didn't put a nucleus around these chromosomes because we're still going to be moving them around. We got a second round of cell division, so why put nuclei around these chromosomes when we're going to be moving them some more? So I didn't put nuclei around those chromosomes for a reason. So now we're going to have 2 cells. One right here, one right here, and they're going to kind of just still be floating around. Okay, no specific spot for these as of yet. They're just floating around in the cell. They got nowhere to be quite yet. They haven't been organized yet. And then I'll put this one over here. Okay. Next, we got to start lining them up in the middle. So I'm going to do this. I'm going to stack them like this. And then one X is going to be on top. I'm sorry if this is taking forever, but I want to make sure we're doing this in detail. I want to make sure you guys get a good review for the test. And then the other one is down here. And then we also have this guy right here. Okay, so lining up in the middle. And then I get to do the fun honors of starting to split these guys up. What a hassle. So, your cytoskeleton, just like up here, is going to start ripping them apart, but instead of having them being ripped apart and moving as X's to each side, now we're going to rip apart the X's into individual strands. So, I'll get these into individual strands for you. So, one is going to look like this. And then that one is going to look like this. And then we're going to have one that looks like this. And then one close. Kind of takes some thinking if you were to do this. This takes a little bit of thought to do this right. And then a short. And then we're going to get a long and then a short. And at the very end, you're going to produce two cells that are nearly done with their own cell division, meiosis 2. And these ones will actually start to form their nuclei because they're going to form 4 individual cells and we're done replicating and duplicating. So one is going to look like this. And this. One's going to look like this. And this. One is going to look like this. This. And last but not least, one is going to look like this. And this. Whoo-wee, that was not super fun to do, but this is the thing I'm trying to point out by the very end, okay? If you look at each of these cells, none of them look alike. None of them do. And that's the point. You're supposed to make 4 unique gametes, 4 unique sperm or eggs. So these 2 dividing cells are about to form 4 cells are either going to be turning into sperm or egg. This one here has half a big fuzz and half a normal line, and then it has 1 tiny normal line, okay? So half my mom, half my dad, and then it got 1 copy of a chromosome that was a little version of my mom's. This is 1 big version of my dad's, and I got a small version that has a little bit of my mom's and a little bit of my dad's. This one here has a big chromosome that belonged to my mom, and this small one that had a little bit for my mom, a little bit for my dad. And this one has a big chromosome that has a little bit for my mom and dad, but then I got one of my dad's small chromosomes here. All 4 are different from one another, okay? They all have different combos, and that's the point, okay? You guys do not look similar to your family members, and more specifically, your siblings, okay? Not to get gross, but you guys, you teenagers, were once kids, and once babies, once fetuses, once embryos, once zygotes, okay? You guys were formed from a sperm and egg joining, and you guys don't look exactly like your siblings, unless you're identical twins, and then that kind of ruined my example. But you guys don't look like your siblings, and that's because your sibling was created from a different egg and sperm cell. So they got different parts. They got some of the same parts as you did, potentially, but they got some different combos together. That's why your sibling can look kind of like you, but also can look drastically different from you, okay? Because each of these sperm are going to be unique, or each of these eggs. You're going to get 1 unique egg, 1 unique sperm, and when they join together, you're going to get something really unique, okay? Something really different from what you are, okay? So that's why siblings can look so different from each other. I hope this makes sense so far. Okay, I'm gonna look through my list real quick. It'll take about 10 seconds. Just one. Okay, I'm just gonna review real quick. Take about 2 minutes. So, how are all the ways that we cause these 4 cells at the end to be unique? First is a thing called independent assortment. Okay, independent assortment is when you're randomly just pulling the chromosomes away from each other and you're just shoving them into different cells, and you're not doing it so they're identical. You're not trying to make sure every cell gets an exact copy, an exact identical copy. You kind of just shove them whichever direction. If you notice, I set this up so actually my dad's chromosome, it's mostly my dad's chromosome, went this way, but one of my mom's chromosomes also kind of went that way. And I did that with the small dad's chromosome and the big mom's chromosome. Independent assortment is when you just randomly assort the chromosomes. Random. If it's random, you might get a unique answer. Another way, besides independent assortment, that we cause unique gametes at the end is crossing over. If we randomly just exchange some of these chromosomes and chromosome genetic information between your 2 X chromosomes, between your homologous chromosomes, you're going to get some unique outcomes, okay? Unique combos. So crossing over, switching a little bit of your parts can also cause unique gametes, okay? Last but not least, most importantly, what's the last thing that can also potentially give you unique gametes? Fertilization. Okay, fertilization is when one sperm meets with one egg, they meet and they make a new unique person. You have these unique sperm that was caused from independent assortment in crossing over. You're going to make some unique sperm. You're also going to make some unique egg. And these unique sperm and egg are going to meet and make something that's twice as unique. Okay? Fertilization. The essence of sexual reproduction. But there is one caveat, asterisk. There's another thing that can cause unique cells, and that's mutation. If you get mutated in any way, your cell is going to be different from the others, because if all your cells are the same, but one gets mutated, well, now this cell over here has a different code compared to all these ones over here. This one's unique now. So mutation also counts within that as well. With that being said, I would recommend studying your model, studying your vocab, looking over the cell cycle, looking over this video. I'll see if I can post this as a 48-minute review. But I apologize that's so long, but I wanted to go in detail. I want to make this good for you guys. Go 2 times speed. Go 1.5 speed. Speeds it up. Okay? But go over it. Do the review. I'm sorry for the virtual day. I wish I could be in person so I can go over all this together. But this is probably going to be more handy for you guys because it will be there whenever you need it. Okay? So I wish you guys luck on the test. I'm looking forward to grading them. Please, Meiosis 1, Meiosis 2, mitosis, cancer, chromosome structure, DNA structure. We went over all of it. If you watch this entire way through and you're starting to get it, starting to understand it, starting to study it, you're going to be in a good spot. Okay. Thank you. It is currently 8:16 p.m. I'm going to go eat some boneless wings. Okay. Go bye.